FIH trials or first in human clinical trials is the topic for present video. I welcome all viewers to this video, and in this video we are going to learn about the FIH clinical trials. When FIH clinical trials start, what are the requirements, why these trials are performed, and what are the challenges. So this will be a short video, and I request you to be with me till the end of this video. It will be a interesting topic. So as the name indicate, first in human. So whenever the new molecules are developed, whenever the new drug are developed, and after the preclinical testing on animals, these molecules are formulated into suitable formulation and given to the human subjects. So this first time study in the human subjects is called as first in human or FIH clinical trials. These trials mark the initial testing of a new drug treatment or medical device in human subjects. FIH trials occur after preclinical studies, that is animal testing, demonstrate safety and potential efficacy. So once the preclinical studies are completed, these molecules are formulated into suitable formulation and given to human subjects. The human subjects may be patients or normal healthy volunteers. Key characteristics of FIH trials are to understand the effect of molecules onto the human subjects. The goal is to assess the basic safety and understand the pharmacological profile of the new drugs. Objectives are to assess the safety, to understand the dosing, and evaluate the pharmacokinetics and pharmacodynamics. Assessing the safety, these trials are performed to understand whether the molecules cause unacceptable harm or adverse reactions in the human or not. And that's why to identify potential side effects and their severity. Then another objective is understanding the dosing. So to finalize the dose range which is appropriate in the human subjects. Determine the maximum dose and minimum dose, that is MTD and MED. Evaluating the pharmacokinetics and pharmacodynamics. So how the molecule behaves in the human body, its ADME pattern and its pharmacological activity is studied. So pharmacokinetics and pharmacodynamics are studied in the human subjects. So there are some phases in the clinical trials. As we know, phase 1, phase 2, phase 3 and phase 4. So first in human trials are the phase 1 trials to evaluate the safety and pharmacological activity of the drug molecules. And typically 20 to 100 participants are involved or enrolled. The participants may be healthy volunteers or sometimes they may be the patients depending on the drug molecule. Study designs generally are SAD and MAD designs. Also cohort based designs are there. SAD designs means single ascending dose and MAD design means multiple ascending dose. SAD designs involves participants receive increasing doses to assess safety and tolerability. MAD designs involves multiple doses are tested to further explore safety and pharmacokinetics. Cohort-based designs are there in which participants are grouped based on dosing levels to compare the responses. So these are some of the study designs for FIH clinical trials. Participants may be healthy volunteers, special population, which may be depending on to the drug indication. Healthy volunteers, most phase 1 trials are involves healthy volunteers to minimize variables related to the disease. Patient populations like specific cases are there, rare diseases, cancer treatment. Informed consent, participant must provide informed consent, understanding the potential risk and benefits involved in being the part of this trial. So whichever the person is being enrolled or patient is being enrolled, informed consent are required. Risk mitigation in the FIH trials. So what are the risks and how to mitigate those risks? Safety monitoring, adverse event reporting, and ethical oversight are the risk mitigation in the FIH trials. Safety monitoring involves rigorous monitoring of the participant during and after treatment with frequent safety checkups. Adverse event reporting, continuous documentation and reporting of any adverse event, immediate interventions if necessary. Ethical oversights, trials are closely monitored by ethical boards to ensure patient safety and compliance with the ethical standards. And this risk is there in the FIH trials because the new molecules are being tested. Before this study, the new molecules are only tested in the animals. And this is the first in human study. That may be the clinical trial 1 or clinical trial phase 1. So some risks are always there. How data from the FIH trials inform later phases. So go no go decision. Whether you are having the satisfactory data with FIH trials or not, that form the basis whether you are going to ahead for phase 2 or not. Dosing guidelines regarding the maximum tolerated dose and the side effects helps to design the strategies for subsequent trials. Pharmacokinetic and pharmacodynamic data generated in the FIH trials provide you the early findings which are crucial for optimizing the drug's formulation and testing in larger population. Challenges in FIH trials. There are various challenges because these are the new drugs which are being given to the volunteers or human beings first time. So there is always an uncertainty that some adverse effect may happen. Unexpected outcomes may occur. Safety concerns are always there. Some unpredictable reactions may happen. Ethical dilemmas are there. Some ethical issues are there for toxic drugs. Recruitment challenges are also there because these volunteers are required to be enrolled and it is difficult to enroll the volunteers which are healthy or which are patients. So recruiting the healthy volunteers and patients for FIH trials can be difficult due to perceived risk. We don't know what will happen in the FIH trials because all these new drugs are there. These are never tested in human beings. So whenever you are doing any testing or any trial first time, so there are always challenges. Coming to the importance, so as it is first in human trials, it provides specific data regarding dose, safety, efficacy, pharmacokinetics and pharmacodynamics. That is the importance. So it is critical for development and it forms a stable foundation for future research and it is a pathway for medical innovation. For any new drug, the FIH trials are necessary to go further for the next clinical trials. So first in human trials, or you can call it as phase 1 clinical trials. In this trial, the drug molecule or newly developed drug molecule is given to the human beings at a very first time. I hope this video will help you to understand this topic and let's meet you soon with such kind of informative video. Thank you.